Welcome back to Biology Domination Crash Course, Day 26. Today, we're entering one of the most important chapters for NEET 2026, Principles of Inheritance and Variation. This is genetics, and if you've been waiting to understand why children resemble parents, why some diseases skip generations, and how traits are transmitted, today is the day everything clicks. This chapter carries heavy marks, and we are going to own every single concept. Let's begin. Gregor Mendel, the father of genetics, chose the garden pea plant, Pisum sativum, for his experiments, and his choice was genius. He selected 7 pairs of contrasting traits, what we now call characters: seed shape, seed color, pod shape, pod color, flower position, and plant height. Each had 2 clearly distinguishable forms. Pea plants self-pollinate naturally, making controlled crosses possible. Mendel studied 14 true breeding varieties over 7 years. This wasn't luck. This was precision. Let's do the monohybrid cross, the foundation of Mendelian genetics. Mendel crossed a pure tall plant, TT, with a pure dwarf plant, small tt. The F1 generation? All plants were tall. The dwarf trait disappeared. Mendel called the expressed trait dominant, here tallness. The hidden trait? Recessive, dwarfness. Now, when F1 plants self-pollinate, TT crosses TT, the F2 ratio gives us 3 tall to 1 dwarf. This is the phenotypic ratio. The genotypic ratio is 1 TT to 2 TT to 1 small tt. This is Mendel's first law of dominance. One allele dominates over the other. Remember, genotype TT and TT look the same, but they are not the same. Now, the dihybrid cross. And this is where NEET questions love to trick you. Mendel crossed plants that were pure for 2 traits: round yellow seeds, RRYY, with wrinkled green seeds, RRYY. F1, all RRYY, round yellow, because round dominates wrinkled, yellow dominates green. Now, F1 crossed with F1. The F2 phenotypic ratio is 9 to 3 to 3 to 1. This is Mendel's second law of independent assortment. Genes for different traits are inherited independently of each other. But, and this is your NEET trap, this only holds when genes are on different chromosomes. If genes are linked on the same chromosome, they do not assort independently. Ratio changes. Remember this. Mendel's laws are the foundation, but real genetics is more complex. Let's cover the key exceptions. First, incomplete dominance. In snapdragon flowers, cross red with white, you get pink in F1. Neither allele fully dominates. The F2 ratio here is 1 to 2 to 1 for both phenotype and genotype, unlike Mendel's 3 to 1. Second, codominance. Both alleles fully express, like in AB blood group, where both A and B antigens appear on the same red blood cell. Third, multiple alleles. ABO blood groups are controlled by 3 alleles: IA, IB, and I. Any individual only carries 2, but the population has 3 options. IA and IB are codominant. Both are dominant over I. This is high-frequency NEET material. Sex determination in humans. Females are XX, males are XY. The father determines the sex of the child because he contributes either X or Y. Now, sex-linked inheritance. The X chromosome carries many genes. The Y chromosome carries few. So males who are XY are hemizygous for X-linked genes. They show the trait even with 1 recessive allele. Classic examples for NEET: color blindness and hemophilia. Color blindness is X-linked recessive. A carrier female, XCXC, looks normal but passes the recessive allele to sons. A son receiving XC from mother and Y from father, XY, is colorblind. Haemophilia A involves deficiency of factor VIII, also X-linked recessive. In NEET pedigrees, when you see more affected males than females and the trait skips generations through females, think X-linked recessive. Genetic disorders. NEET tests this heavily. 2 categories, chromosomal and Mendelian. Chromosomal disorders result from abnormal chromosome number, aneuploidy. Down syndrome is trisomy 21, 3 copies of chromosome 21, total 47 chromosomes. Features: mental retardation, short stature, characteristic face. Klinefelters, 47XXY, male phenotype with feminine features, infertile. Turners, 45XO, female phenotype, sterile, short stature. Now, Mendelian disorders: sickle cell anemia, autosomal recessive, single nucleotide mutation in beta-globin gene causing glutamic acid to valine substitution. HbA becomes HbS. Huntington's chorea, autosomal dominant, progressive neurodegeneration, appears in middle age. And remember pleiotropy, 1 gene, multiple effects. Sickle cell gene affects shape, oxygen carrying, and cell lifespan. Let's lock in today's wins. You now understand Mendel's 3 laws: dominance, segregation, and independent assortment. You can solve monohybrid and dihybrid crosses with confidence. You know the exceptions: incomplete dominance with its 1-to-1 ratio, codominance in blood groups, and the 3-allele ABO system. You understand sex-linked inheritance, can read pedigree charts, and know the difference between chromosomal and Mendelian disorders. This is NEET-level mastery. Day 27 is molecular basis of inheritance. DNA structure, replication, transcription, the molecules behind what Mendel saw in pea plants. Subscribe so you don't miss it. Drop a Punnett square in the comments if today clicked for you. See you tomorrow. Keep dominating.